And now I'm super thrilled to introduce another speaker who's going to talk about a slightly different version of this problem, which is something that at Madrona we're super passionate about. And that's how do you combine really innovative and high throughput wet lab biology with machine learning in interesting ways, especially as it relates to therapeutics or different life science and biotech applications. So I'm thrilled that I get to introduce Geoff von Maltzahn. Geoff is a general partner at Flagship Pioneering, which is the innovation and incubation studio in Boston behind companies like Madrona and Sana here in town. He's the inventor and founder of, I think, at least 10 different companies over the last 12 years, including Generate Biomedicines and Therapeutics. And it's just going to be a really, really exciting and awesome talk to hear what they're up to and what he gets to do every day. So without further ado, Geoff.
Awesome. Thank you, Chris. So I'm not going to spend a lot of time on my daily routines, but I'm going to spend a little bit of time in this realm. And just a few moments of glancing at this slide are probably evidencing what we in the field of biology have felt for a long time, which is our human brains aren't great at understanding biology. And yet this code, of which we're born with millions of slides of, 3 billion base pairs from each of our parents, explicitly encodes the quantitative function of every single motor, every machine, and the grandeur of what we call life. And the reason this is pertinent to this conference is that with the help of a few brilliant machines, we're starting to actually be able to read this code. And of course, we've made amazing progress sequencing this code, but it's sort of been gibberish relative to reading and comprehending. And we've started to make progress in writing this code. So I'm going to give you a couple examples of that. But the broader implications of biology transitioning from an era where we've been more or less been practicing guesswork to being able to predictively succeed are as important as the other transitions of scientific subjects that we learned about in high school. So if you rewind 150 years, every time a topic in science that we learn about in high school has transitioned from expensive tinkering and trial and error to predictive success, the entire world has changed. So a subset of chemical systems doing that changed every single industry. A subset of electrical systems doing that changed every single industry. And we're in the midst of the beginning of that transformation in life. And so any chunks of life making that same transition are going to be a really, really, really big deal. And it's ever more evident that machines are going to be a driving force in this. So what we work on in Flagship is trying to invent new technologies and start companies. All the companies that we're starting these days are from science that we're practicing internally. And if you're in a field that's plateaued, you would be wise to stick to more or less the bullseye of this slide, either stuff that already works or slight improvements upon them. But if you're in a field that is changing very rapidly, it's amazing how often there are these outliers where the risk associated with a new pursuit might be comparable to doing something incremental, but the implications of success are extraordinary. So those are the things that we try to exclusively focus on. If somebody's already working on something, we will shut down or avoid or try to venture further from shore in pursuit of these rare scenarios where if something works, it has a chance to change the world. So I was trying to think of ways of making a more visual slide than that, and I chose this one from your local Seattle Art Museum. And this is a depiction of Puget Sound, and I think it's beautiful. But what I love more is in looking into it, it is suggested that the artist had never been there at the time that they painted this. And so, in fact, the beauty in this painting is the beauty that one is imagining at the start of any of these voyages to go and try to do something really special. And biology is going to be full of voyages akin to the era of great exploration where people were lucky enough to get to sail around the world. But we're going to get to figure out how biology really works. And everything that we work on starts with a question that can be formulated as what if, ... And it's meant to be a supposition of if something was possible or if something was right, it might just change the rules that we think a given area of medicine or category of life science operates based on. And I'm going to give you a couple examples across the information flow of life. So DNA becomes RNA, becomes a sequence of amino acids which then folds itself into the technology that performs a desirable function as a therapeutic or within our biology. And Flagship's most famous for the what if that became Madrona, which was, what if you could deliver any mRNA to any cell in the body and produce any protein as a result? I'm going to start with an example upstream of that. So in the code of life itself, and trying to create machines with the ability to write a single letter or write thousands of letters into desired locations. And then we'll end closer to the right side of this. So as I mentioned at the outset, as a field, we've made astonishing progress in sequencing the code of life. We had one human genome 20 years ago. We have millions today, and that is headed towards billions. Concurrently, we have made a lot less progress in writing that code. So a case in point is if you're a child born tomorrow and you are missing the 10 pink letters on this slide, you probably wouldn't get to see your first birthday. But if we could write those letters into exactly that location, they'd get to live a normal life, and they might not even remember the name of the disease that they were born with. That is the defining aspiration of what we're working on in Tessera. And if you go back to that information flow of life, you'll notice that it flows from left to right, and our progression in medicine over the past 100 years has been climbing in reverse up that current. So the foundation of medicines has been small molecules that drug proteins. Proteins themselves became therapeutics at the birth of biotech. Hopefully most people in this room have experienced the benefits of mRNA vaccines over the past couple of years. And now the first cures are taking place at the level of DNA. And if you're trying to cure a disease or prevent disease from ever happening, DNA is going to be the rightful home for many of those therapies. And we are about 10 years into a revolution that was kicked off with the machinery that you're seeing here. So this has the wonderful acronym CRISPR. And it is an incredible pair of scissors in nature that chops up invasive DNA. And 10 years ago, its action was decoded and it was introduced into human cells for the first time, showing that, in fact, you could more or less program on a sheet of paper an RNA that would direct this to go to a specific place and cut it in half. Our cells don't like having the genome cut in half, so they swarm in to try to repair it. They often make an error in the process which turns off that gene. For therapeutic applications, in a rare number of scenarios, a mistake pre-written into DNA can cause a gene to become toxic, and then turning that gene off has an enormous amount of therapeutic value. But most of the time when mistakes happen in biology, the things break down. I mean, it's a lot like when technologies break in your house. And if when things break in your house, you could only unplug them, most of the time you'd still be left with a problem. So to fix things in biology, you typically have to be able to write a short sequence or a long sequence into a desired location. And the start of Tessera dates back to asking this question about 7 years ago, which is, well, if CRISPR evolved to be an extraordinary pair of scissors, let's go looking for machinery that might have spent billions of years learning how to write. And in fact, we realized that even asking the question might have been insulting to Mother Nature. Of course, she invented this. And they're kind of like the smallest circle of life. So if you imagine moving just one step up from a virus, what these are mobile genetic elements, and they are sequences of DNA that encode one or more proteins that can grab onto a copy of themselves and then write that into a new location. And like many areas of biotechnology and biology that have the chance to change medicine, this one has an amazing scientific history, in that the first of these was discovered before the 3D structure of DNA was even known by Barbara McClintock. She had the bravery to squint at gels in Cold Spring Harbor and imagine that a sequence of DNA must have the ability to encode machinery to allow it to hop from one place to another, thereby regulating the diverse pigments that you're seeing in corn on the right. In her words, her hypothesis was met with confusion and hostility from colleagues. In fact, she stopped presenting on the subject for a period of time. She was experimentally validated decades later and received the first unshared Nobel Prize in Medicine and Physiology for a woman in the 1980s. And now we know that that first life form that she had the brilliance to dream up is one of a category of machinery that are all over the place. So about half of every one of our genomes was built by mobile genetic elements hopping around over the past few billion years. Similar chunks of the animal kingdom, plant kingdom, and beyond. So our question transitioned from, what if there's machinery out there that does this, to, oh my God, where do we even start? And particularly, which one of these still work and didn't break down millions of years ago? Of the ones that work, which could work on the human genome? Of those, which could we program so that this machinery could write, a therapeutic sequence into a desired location instead of themselves, which would be inserting something that could then hop around and jump to new locations? That would be a bad idea. And when we can computationally predict those parameters, we do. But in many scenarios, we had to learn the rules by which these operate by building the kind of platform that Chris described, a means by which we can conduct very high-quality experiments in high throughput and continually learn the rules by which these machines function. And as a testament to the era of biology that we are all in, we've been able to synthesize over 40,000 distinct gene writers, test them in human cells, and we've been learning from all of this. And we've now automated a lot of this, so that the rate at which we can do these experiments is going up all the time. And the result of that to us has been pretty spectacular, in that mostly as a testament to Mother Nature's ingenuity, we've been able to create gene writers and optimize them with machine learning that can make each of the modifications you see here. So gene writers that can change any letter to another, so an A to a T, A to a G, A to a C, and vice versa. That on its own would provide the ability to cure most genetic disease on a patient-by-patient basis. Below that, programmably inserting and deleting, and then we've sort of doubled down on the ability to write entire genes, so thousands of letters into a desired location. We're working across every major biochemistry of mobile genetic elements, and those create what we call RNA gene writers, which utilize the machinery of what are called retrotransposons in biology, and DNA gene writers, which utilize the machinery of the two other major classes, transposons and recombinases. RNA gene writers turn out to be engineerable to make each of these modifications with different types of gene writers. And DNA gene writers are probably exclusively well-suited to the bottom. In fact, we've got some that in nature insert 100 kilobase sequences. That's 100 times larger than the average human gene. So you'd really have to dream to want to do that in human biology. And I'll show you here an example of how these work. And you should contrast this with CRISPR doing its thing and chopping the genome in a specific location. Here, an RNA gene writer grabs onto an RNA that we administer, goes to the genome. It recognizes a specific location and nicks one strand. That allows the RNA to anneal to that site. If that happens in a sequence-specific fashion, then it activates a reverse transcriptase domain, which one letter at a time writes a DNA sequence corresponding to that RNA strand. It's then able to go over and complete the second strand synthesis, resulting in a new piece of DNA, letter by letter, written into the human genome. So this is an advantage of letting Mother Nature spend a couple years, billion years with a head start on a problem. Like, this life form, so to speak, wouldn't have gotten to survive if it hadn't figured out how to solve these steps autonomously. But the natural ones don't work as well as you would like in the human genome. So we've had to learn a lot of the rules by which these function with the benefit of machines and the platform we've built, so as to enable them to perform therapeutic feats. And this is an example of one of those. So if you were a child born with a T in this location, or born with a T in this location from both the DNA from your mother and your father, that would prevent you from metabolizing an amino acid that is essential for all of us, and in fact is present in all protein. So if you had two Ts, even a single slice of bread might overwhelm your body's capacity to metabolize this amino acid, causing it to accumulate to toxic levels, which can lead to severe neurotoxicity or worse. This is an important indication for us for two reasons. One is we've been able to change that T with what to us is remarkable specificity and efficiency. And the second is that a member of our team's son, Jacob, has one of these Ts. And as his parents would say, whatever your hopes and dreams when your children are, when you're expecting children on the day of their birth, you're really just hoping that they're going to be healthy. Jacob's lucky in that he has one T, not both, but his levels of this amino acid are always 10 times higher than those of us in this room would have. He's also lucky in that his grandmother is an MD, PhD pediatrician geneticist, which is not always the case. So he has, from the moment of diagnosis, benefited from some of the best care that is possible. So for any patient with one or two of these Ts, if you could make the correction back to normal in 10% of the cells in their liver, you would provide a lifelong cure for this disease. Our gene writers are now operating at about 5 times that level of efficiency in the preclinical models that we're working in. And this category of medicine, in contrast to most areas of medicine, has remarkably high translation from preclinical successes to those successes manifesting in the clinic. So that is just one of the things that we're working in. And the category of gene writing evidences some of the principles that the future of therapeutics of this kind are going to have, and that companies like Madrona have had the benefit of. In that, at the beginning of Madrona, Stefan had often said that there was no version of Madrona that would have a drug. It was either going to be 0 or a whole bunch. In that, mRNA being the center of the central dogma, it wasn't going to work, in just some tiny corner of medicine. And similarly, if we're successful in being able to make a change like the one that I just described, the probability that we're going to be able to do that for a wide variety of diseases in the liver caused by single-point mutations goes way up. In fact, we've already shown the ability to be able to preclinically cure a number of distinct indications. And similarly, if these gene writers can work in cell type A, and we can devise ways of delivering them to cell type B, the probability that they're going to be able to create a whole new category of medicine in each of those cell types goes up as well. And within the company, we've invented ways of being able to deliver gene writers to hematopoietic stem cells, the HSC category. For example, if you could change the one base pair that causes all of sickle cell disease in 20% of the stem cells that live inside of the bone marrow, you would provide a lifelong cure for sickle cell disease. And the category of RNA gene writers I just described offers the potential for something like a COVID vaccine formulation to make that possible. An injection of lipid nanoparticles that goes to the bone marrow, writes that one letter back to wild type, and provides the possibility of a lifelong cure. So I'll give one more hint of how vast the future of genetic medicine can become, and it's embedded in the middle part of this, in that all vaccines, mRNA included, work by instructing a subset of your cells called B cells to change their genome and produce a protective factor called an antibody thereafter. As we gain the ability to be able to write into cell types across the body, the capacity to prevent disease from ever happening is going to go way beyond infectious disease. And particularly, as we've gone from sequencing one human genome to millions, it's already become evident from genome-disease relationships that if you could write the 3 letters on the left into that location, you could provide vaccine-like protection against somebody ever having a heart attack. People with this allele or comparable have about an 80 to 90% lower probability of having a heart attack from everyone in this room that isn't fortunate enough to have that modification. On the right-hand side, if you could write both of these Ts, you could provide vaccine-like protection against ever having Alzheimer's. So, future of this category, powered by machines helping us to be able to invent therapeutics of this kind, may just offer us medicines that prevent diseases from ever happening, for which have eluded our ability to reverse or even mitigate to date. So I'm going to jump to the right-hand side of this and move quickly, in that the information flow from DNA becoming the engines and motors and machines and sensors and devices that make life possible has sort of been a mystery. Like, all of the quantitative functions of every protein are encoded by the DNA sequence that they come from. But we haven't had any mastery of those relationships to date. In the center of this, the linear sequence of amino acids folding themselves into a 3-dimensional structure has had extraordinary advances over the past 2 years, including today's meta announcement, which was discussed earlier. We set out about 5 years ago to try to crack the bookends of this, to say, well, gosh, the information must be there in DNA. And so what if we could learn everything it means to be a protein with a given function, and maybe everything about a quantitative function from DNA itself and from every piece of information we can get of DNA structure all the way to proteins. And at the time, that sounded crazy. And we interact with a bunch of people in various fields, and mostly the suggestion that this was going to work was that we were not going to be using our time wisely to even try. So in particular, if you were going to pick one function in all of medicine to try to master for proteins, it would be pretty easy to make a list in that all antibody value, all therapeutic, all cytokine, all growth hormone, and all peptide value is based on one function. It's the ability to stick to a desired target in a very specific location while not sticking to other things. And if you could master the functional rules governing that, then the origination of medicines across every one of those areas changes completely. And in fact, most proteins are of an average size of 100 amino acids or so. Every one of those spots, you have 20 options. So the combinatorial space of proteins is 20 to the 100 power. It's like atoms of the universe squared. It makes it evident why it's more likely that every single therapeutic that's ever been approved within the realm of antibodies is suboptimal than that it is optimal, given that nobody has had all the atoms of the universe to conduct their discovery campaigns for these. So often I have to explain this to audiences, but I would almost be insulting you to do so. None of these people have ever existed, and statistically they never will. And we thought about applying the same premise to proteins, not to just sequence to structure relationships, but particularly trying to master the rules associated with protein binding events as a function and other functional parameters. And the basis of Generate was the thesis that the way that amino acids like to interact with each other inside of proteins probably has correspondence to the way that they would like to interact with one another on the outside of a protein. And therefore, we've learned from every crystal structure of every individual protein so as to try to extrapolate those relationships to generate therapeutics. But additionally, all of the protein-protein interactions, all of the antibody-protein interactions that are known have gone into our database. And in addition, we've learned from the raw sequences of hundreds of millions of proteins in DNA that has been sequenced to date. And this was all based on the idea that this could allow us to, instead of conducting a discovery campaign, if we wanted to generate a binder to a desired site, might just be able to say, what is the DNA sequence encoding an antibody that would hit this exact epitope in this binding pose and with the following affinity? So all of that public data allowed us to kind of start to crawl and maybe walk. But as Chris described, we've put a lot of effort into building an integrated platform inside the company such that every model that we're using is able to generate a set of hypotheses that are built and evaluated in-house, and so that every future model benefits from every single data point taking place across every single campaign and every separate program that we're working on. And that has allowed us to do some things that are pretty incredible. And it's probably the basis upon which future biotech companies are going to create value and impact while expanding their capacity to discover along the way. To date, scale has been the enemy of corporate ability to be able to discover in biology. That's been the home of small teams. And it is likely to become the friend. And here's some examples of things that we've been able to do. I'll give you an example in the next slide of de novo prediction of an antibody to a novel epitope. So an epitope that, as far as we're aware, antibodies in immunology and biology aren't able to hit, and we've been able to generate multiple antibodies against those locations. Next to that, almost on a whim, I asked the team to take the top $50 billion of antibody therapeutic sales and see whether we could generate sequences that fell outside of the intellectual property of those parent antibodies and bound to the exact same epitope in the same binding pose with a higher affinity and other desirable attributes. Within 3 months, we were able to do that for 100% of them. If you had all of a large pharmaceutical's current discovery approach, you probably wouldn't be able to do that in 5 years. In that you'd be finding things that stick in random locations and novel orientations, and you wouldn't find one that hits the exact site that you're interested in. When we've shared that with big companies, sometimes it's met with an expletive. In the middle here, we've been able to take take non-human proteins from bacteria, and rebuild them so they're entirely composed of 9 amino acid sequences that are either already present in us or are hard for our innate immune system to recognize. To do that, we've had to change 60+% of the protein sequence, in some cases 70+% of the protein sequence. So this is a couple hundred protein amino acid. It's called asparaginase. That's like atoms of the universe cubed. And with roughly 50% probability, the ones that we predict across those vast changes in sequence, in fact, still function as an asparaginase. And some of them, when we predict up a functional gradient, function better. It's like jumping off Earth and landing on another planet with even nicer weather outside than we're enjoying today. Statistically, that would be impossible. And when you dose these into mice, in fact, it's very difficult for them to be able to generate antibodies against them. And the implications of that for AAVs and cell therapies are pretty vast. Here's a little highlight of one of the de novo campaigns we've been running, which is all of us know that from SARS-CoV-2 that though Mother Nature is a wonderful friend, she is an awful enemy. And the evolution that the new variants have continued to undergo in the receptor binding domains has more or less rendered all of the approved antibodies useless, and many of the antibodies that we've been generating from vaccinations. There are locations, though, that perhaps because our immune systems aren't good at making antibodies against them, have almost remained constant from the original COVID to now. And we've been able to generate antibodies against those domains. They're on the S2 stem helix and on the fusion peptide domain that are able to neutralize every single one of the variants that has come out to date. So that's a program we're going to be moving into the clinic next year. As far as we know, it'll be the first completely computer-generated antibody. And from our perspective, in the not-so-distant future, all therapeutics are going to be generated that are in the form of proteins. And it's probably the case that in the not-so-distant future, similar themes of transition from trial and error and human intuition and nuggets of wisdom are going to seed terrain to the brilliance of machines. And so I'll leave you with the premise that we are likely at the very, very beginning. It is hard to be simultaneously amazed by things like the COVID vaccines reaching more people than all protein therapeutics combined in just the past 2 years, and yet realize that we are just at the very, very start of this transition of generative biology and a frontier of predictive success in life sciences. So look forward to many, many continued conversations along those lines. I'm really grateful to be here today. Thank you all.